Okay, so I was an undergraduate in Boris's lab from 1972 to 74. And unlike all the other people who were undergraduates, I was actually an MIT undergraduate. And I was not a tutee of Boris's because MIT didn't have that program, but I did interact a lot with Boris. And the reason I actually came here is because Boris had a very important impact on me. I think the spirit of Boris's scientific spirit lives on in me still. And a lot of people have commented on many of the aspects of that. One thing that was from the beginning is that after I'd been in the lab a little bit of not too long, he was Boris. And that's not something I was expecting really to have to talk to Boris and call him Boris when I was like a 19-year-old kid. And I think that actually really conveyed an attitude that was very important. Everybody's talked of the thing that I really appreciate is the scholarship, the intellectual aspect of it, the rigor of it, the creativity of it, focusing on the main point and keeping everything simple. Everybody said that. There's one other point that hasn't been brought up, which I think is extremely important to me, is that Boris was a systems biologist in that he actually, although he was really a microbiologist and that was his real genius, I mean, the guy could remember, the standing joke was if you gave him a piece of paper, he could reproduce the entire metabolic chart himself. But he also did quite good genetics in the lab, there was good biochemistry in the lab, and actually, really it was solving the problem. And whatever technique you did to solve it was good. And really certain techniques were good for some things and others for other things, and that's a really important lesson. And at least early on in my career, especially when I went to, as at MIT, as I said, which was a very genetically oriented place due to Boris, my graduate work was at the Stanford Biochemistry Department, which was exactly the opposite. It was an extremely and really high quality place, but very biochemical. And I kind of spanned between those, and I think that was really important, and I've continued that pretty much my whole career, at least I tried to. I want to thank one other person. You see Albert DeLeo on here. He's unfortunately not here. Albert was a postdoc during my time, and he was the guy who really taught me how to actually do an experiment. And another thing that was nice about Boris for me was that he really let me do my own project. So I really had my own project that no one else was doing. And Albert was incredibly nice, showing some crazy undergraduate how to actually do an experiment and do controls and things like that. So the first thing I'm going to mention in my talk here is something that I've never talked about in all the talks I've given. This is the work I did in Boris's lab. I've never actually talked about it, so I'm going to do it now. So I basically, for reasons that probably made no sense really, I wound up isolating an ammonia-sensitive mutant of Klebsiella aerogenes. And this was a mutant that would not grow under normal conditions. Actually, it was a TS mutant, but under the restrictive conditions, it wouldn't grow under normal conditions, but it would grow if you dropped the ammonia concentration by a factor of 30. And that was a new phenotype. And we're trying to figure out what it did. And one of the ideas I had, well, what could I do to make it grow? So I went to all the freezers that were in the lab, and I took every conceivable compound and tossed it in there and said, what will make this thing grow? And it turned out a bunch of things made it grow. And so I sat down with a metabolic chart. It took a while to figure this out, and eventually I sort of figured out that the only thing that made any sense was that it was defective in making succinyl-CoA. And I then found out, that I did an enzyme assay with Albert helping me out, knowing to do it, that in fact the mutant was a temperature-sensitive mutant in alpha-ketoglutarate dehydrogenase, which is why it was not making this, and it was actually about an altered KD, which is why it was a problem. And it turns out that the reason why it was an ammonia-sensitive mutant is because alpha-ketoglutarate lies at the intersection of carbon and nitrogen metabolism, and there's a competition for the alpha-ketoglutarate by the nitrogen pathway and the carbon pathway. And when you're a little bit messed up over here, you can have a problem. And it turns out in high ammonia conditions, normal conditions, glutamic dehydrogenase is an incredibly potent active enzyme, and it basically sucked out all the alpha-ketoglutarate, causing the problem. Whereas, if you put it in low ammonia, this basically wasn't made anymore, and you had glutamine synthetase, which was essentially doing it, but it couldn't compete well, and so that's why it would grow. And if you grow these cells under conditions like citrate or pyruvate, where you have much more Krebs cycle activity, that also bypassed the whole thing. So that was quite an amusing thing as an undergraduate project. Actually, I wound up getting a master's degree for some useless reason based on this. And it turns out that I'm very proud of this paper. It's been cited 5 times. The first time was a paper several years later on Antarctic yeasts. And the last time was last year, which is 38 years later. And I looked, of course, I looked up these papers as soon as I saw them, and they all cited it correctly. So anyway, that was my lab experience in Boris's lab. Okay. Now, when I went to Stanford, the biochemistry department, that's where recombinant DNA was basically beginning, and I was one of the first people to really do recombinant DNA, as John undoubtedly remembers. So anyway, so the very first thing I did, that was the time where cloning was just starting and doing functional selections. And so this is the first cloning of a gene by recombinant DNA technology that was a non-E. coli gene. And you could see what organisms I was using. That came directly out of Boris, those organisms. This is the old days. This paper came out 5 years after I actually did the work. JMB. That's why it no longer exists as a journal, really. Okay, so the other thing is that when I left here, I left MIT as an undergraduate, and I went to Stanford, the thing I really wanted to work on was eukaryotic gene regulation. And I was particularly inspired by this comment, a famous comment by Jacques Monod, which is, what is true for E. coli is true for the elephant, only more so. And so I really wanted to work on gene regulation in eukaryotes, but at the time there was virtually nothing known, other than there were 3 RNA polymerases and there were mutants like Gal4. That was about the sum total of all knowledge of eukaryotic gene regulation. So the way I went out to start this was to actually clone a gene from yeast, it turned out, by functionally expressing it in E. coli, and subsequently showing that the enzyme encoded for the His3 gene. And basically this came right out of my Boris experience, because this was P1 transduction done with recombinant DNA technology. And so the idea was to make a library, and the amusing thing here is that these things that are now called libraries were called libraries at Harvard, but at Stanford they were called pools. And in Switzerland, they were called banks. And this is actually but anyway, Harvard, wins all the time, so they're now called libraries. So anyway, so this was actually the first expression of eukaryotic DNA E. coli to make an actual functional protein. There were some very interesting times with the politics. John referred to them. We were sort of on opposite sides at the time, because I was practically closed down, because this was expressing eukaryotic genes in E. coli, which was considered like not a good thing to be doing. Anyway, this is actually the original experiment, and this is one of my favorite. This is my favorite experiment I ever did. This is the original slide back in the days when I used to hand draw them. So this was an experiment where you take this hybrid pool and it complemented this mutation, and you got a fragment of DNA that expressed the gene. Then that basically, I did eventually show that the enzyme that got made was the right enzyme, but the really key experiment that showed genetically that this was the right gene was I using a technique that was called Plaque filter hybridization, this ancient technique, which was brand new at the time, invented in Ron Davis's lab where I was a student. I cloned the exact same fragment from a mutant, and actually 2 mutants, and the 2 mutants, if I cloned that gene, it would not complement. But if I crossed them as a phage cross, it would complement. And in addition to that, I also cloned one from an AMR mutant, and that wouldn't work unless I put in a suppressor tRNA. So you can see that those experiments are Boris experiments. And I should point out that I was in a department of biochemistry with Arthur Kornberg, and that they weren't thinking that there was only one geneticist in that whole department. A guy named Dale Kaiser. I recognized the last name. I was the father of Chris, and I think the first time I ever met you, you were still in high school, along with Chris Hogness, who was. So anyway, so that led to basically an approach we call reverse genetics, which you have a clone gene. So instead of going the classical way with mutants and all the usual way, you start off with a clone gene, you do the usual molecular characterization. I shouldn't say usual, I did my first Southern blot off a preprint from Southern. Northern blotting was then invented in the labs next to me. But the real essence of this was to take the clone gene, manipulate it by making deletions and later other fancy gizmos, and to put it back into yeast. Thank you, Jerry Fink, for discovering how to do yeast transformation, and then do phenotypic analysis. In this way, you could basically do structure function thing. And that was, again, very much a combination of biochemistry, genetics, and that is no question was influenced from Boris, because it certainly wasn't coming from the Stanford Biochemistry Department. Okay, so I'm going to talk about what have we learned since that time when I got into this about eukaryotic gene regulation that's worth knowing about. How is it different than bacteria? And this is the prokaryotic gene regulation a la Jacob and Monod plus Potashny Gilbert and all the early guys figured this out. And basically, the concept is you have a promoter, which basically is recognized by RNA polymerase, which recognizes the sequences and makes transcripts. There are two forms of regulation. The Jacob-Monod type is repression, and you have a sequence, DNA sequence called the operator. The repressor protein binds to it, and usually it binds right around where the promoter is, and so it just basically blocks everything, and it doesn't work anymore. And that is a repressed case. Now, Jacob-Monod didn't like this, but eventually it became clear from Ellis Engelsberg's work, a guy who has been completely forgotten in the history of gene regulation, which is ridiculous, that in fact there was cases of genes being activated in where you have an activator protein binding to I'm going to use the word enhancer, and it stimulates transcription of the polymerase. And in this particular case, the activator typically works by actually directly interacting with the polymerase. And in fact, this activator is often really close to the polymerase and directly touches it, but it also can work pretty far away. And one of the main discoverers of that was Boris Magasanik himself and some of the people here who discovered that at least some activators could work quite a distance away. Okay, so that is the bacterial case. So I'm imagining Boris in front of me right now. And Boris, I'm telling Boris this, and I actually did have a conversation like this, Boris would say the following thing. So what is new? So one thing that you could imagine is that nucleosomes coat the DNA and inhibit the access of transcription factors. That's clearly a eukaryotic thing that bacteria don't do. But the question is, does this actually matter? Okay, so there was a school of thought, Mark Potashny said it nicest, is that nucleosomes are high salt. And so, yes, they may make a mess of everything, but what difference does that make? It's just harder. It doesn't really tell you anything different. So that's one issue. And the next issue is this, that's different. And that is that most DNA binding proteins have a relatively limited specificity. It's 4 to 5 logs, which is very impressive when you look at a biochemical experiment. But it's not so impressive. That specificity turns out to be about 7 or 8 base pairs of information. And if you're in the E. coli genome, you have 4 million base pairs, so that's not that great. And if you get to yeast, you're now talking 10 million base pairs, that's worse. And if you get to the human genome, you're talking 4 billion base pairs. So you've got a problem here, which is that this specificity is not really all that good. And that means there's a problem, and that bacteria don't have that problem as much. So that's something one has to think about. So what is new? So one thing that is new is that we have enhancers, we have activators, we have operators and repressors, we have transcription machinery, so that's not new. One thing that's new is the transcription machinery is vastly huge. It's not RNA polymerase with 4 subunits. It's actually a collection of what are called general transcription factors and mediator. That's, I forget how many, 50, 80, some ridiculous number of proteins. So it's not, the machine is more complicated. But so what? So it's a big complicated machine. That really, conceptually, isn't any different. And the DNA binding activators and repressors, that's conceptually not different. But one thing that really is conceptually different is that these activators and repressors typically do not directly work on the polymerase. Okay? So the repressors don't typically work by just blocking things. Some of them don't work by blocking things. They need a co-repressor that blocks it. So the DNA binding protein, just binding per se, doesn't do much. Okay? It doesn't repress. You need a co-repressor. Similarly, the activators can bind, but they don't work by touching the polymerase. Okay? So that really is different. Now, one case of a repressor that's quite dramatic and a co-repressor is in the heterochromatin and silencing. This is the yeast silencing, which I'll come back to a little bit later. So here is the DNA binding protein called RAP1. That recruits the co-repressor, which is the SERV proteins that Lenny Guarente has done a lot of work on. These SERV proteins actually can self-assemble by protein-protein interactions, and they can form a filament, essentially, and that represses transcription. But the important point is that this repressor, the SIR complex, is not a DNA binding protein per se. It is recruited by a DNA binding repressor. So that is a fundamentally different kind of mechanism than anything that would be in bacteria. And this example is also seen in polycomb complexes and the HP1. It's the same basic idea. Those are co-repressors. Now, another model that we discovered a while ago, and a few other labs did too, is that some repressors work by recruiting a complex of DNA that is a histone modifying enzyme, a histone deacetylase. And so this repressor doesn't work on its own. It must recruit the deacetylase. The deacetylase takes away acetylation residues on the histones, and that is a critical aspect of the repression. Again, there really isn't a bacterial counterpart of that. And a related version of that, the opposite side of that, is for activators. So the activator proteins, again, don't work by themselves, and they don't really directly touch the polymerase. It's a little unclear what they do in the whole transcription machine, probably its mediator. But the other thing that they do is they recruit chromatin modifying complexes. And there are two kinds that it recruits. One is a nucleosome remodeling complex that actually kicks the histones off the DNA, and you create open DNA to make it accessible. And the other thing it does is recruit histone acetylases, which make a big acetylation mark. So that is creating a completely novel region of the genome, and that's a critical aspect of the activation process. Again, the activator is not even working at this level. The activator is not even working on the transcription machinery directly. It's working on the actual chromatin template, which is, again, a very different conceptual model. So I've told you about activators, repressors that recruit a lot of different kinds of repressors, including those silencing things. The other thing that's different is the RNA polymerase itself, the elongating polymerase itself, also recruits all kinds of histone modifying activities. So they're histone methylases, acetylases. The act of this can also result in nucleosome depletion. So the act of transcription itself is not just simply the polymerase going down the gene. It is putting chromatin marks into the chromatin. And so, in fact, the chromatin modification pattern is completely determined by classical Jacobineau mechanisms, namely activators, repressors, and poly-2 elongation, but they're putting in marks into the template that are used for things. Again, a very different concept. And one example of that, which I pick, of course, from my lab, is this particular histone methylase. Does this work? Okay, which puts in lysine 4. And so the idea is when the elongating polymerase can recruit this enzyme and it modifies the DNA. But here's an important point. The way this experiment is set up is we have transcription, we put in the mark, and then we shut off the transcription completely by just throwing in glucose, okay? And the important point here is that at a certain point, there's no transcription anymore, but the mark is still there. And as a result, that provides a localized mark and memory of recent transcriptional activity. So this is, again, a very important point. The template on which the machines work can be different at different times, and you can put marks in where they'll stay there for a certain amount of time, and during that time that they're there, they can actually affect things. There's no bacterial counterpart of that. So that is a fundamentally new concept. Now I'm going to go back to this DNA binding specificity question and tell you what we've done on that. What we found is that the strategies for specificity differ in E. coli, yeast, and human, and these are all experiments. The E. coli genome is permissive, and we did these experiments with Lexay. These are genome-wide chip experiments for those familiar with the technology. The interesting thing is that there's a near universal relationship between what is bound in vivo and what is bound in vitro. That is, the E. coli proteins are looking at the sequence, and that's it, okay? Now that means that, first of all, the E. coli proteins typically have high levels of specificity. They're higher levels of specificity than mammalian ones, because they have no way to deal with this specificity problem. So the way they deal with it is, number one, they have higher specificity proteins. And number two, we showed in the Lexay case that there's actually an evolutionary selection against having the motifs in the wrong place. Because when you have a motif in some random place, it's going to bind there. And so, and if you put artificial sequences anywhere you want, it'll bind there. So that's what E. coli does. Now yeast is different. In yeast, the same sequence isn't always bound. So if you have a binding site that's in a promoter region, your favorite DNA binding protein will bind there very well. But if in the exact same sequence in the coding region, it won't bind at all, or binds 20, 30-fold less. So that can't be explained, that has to be explained by something. And it turns out what we found out in about a 30-year project, it was actually one of my favorite historical projects of what we did in it, is that it turns out that yeast promoters are intrinsically accessible. And the reason they're intrinsically accessible is that they contain DNA sequences that have homopolymer tracks of A's and T's, and those sequences are intrinsically bad for nucleosomes to bind them. And so the nucleosomes don't like those sequences, and as a result, so there's sequence information there, it's just not specificity information. So the sequence information, that information says we don't like nucleosomes. Now proteins can now bind much more easily. What you've effectively done is to reduce the effective genome size. So that's how you deal with the specificity problem. Now this mechanism is only worth about 20-fold, and humans have too much DNA, so this does not work for humans. And in humans, it turns out from experiments that we've done, and many of us have done as well afterwards, is that a very small percentage of the DNA sequence motifs are actually bound in a living cell. And in fact, also a fair number of the actual binding sites don't even have the motif, okay? So that means that human proteins must bind cooperatively. That's the only possible way to get specificity. That you have to have sequence information from more than one protein at a time, because having the motif alone isn't going to, there's no way to determine one site from the other. And if you hear many people talk about this, they said, well, it's because it's open chromatin. Well, that's not really quite right, because the chromatin can help mediate cooperativity, but chromatin has nothing to do with the specificity. And also, if you think about it, if the chromatin is open, how did it get open? It doesn't just magically open up. So it's the DNA binding proteins again. And so the way we think about the cooperative binding is as follows. Now, most everybody knows that the classic cooperative binding is protein here, and they have protein-protein interaction. And that certainly happens, but that can't be a general explanation for this, because there's many, many examples of this throughout the mammalian genomes. And so you need a different cooperativity mechanism. The way we think of it is cooperative binding without direct protein-protein interactions. And the way we imagine it is as follows. We start off with a nucleosome. Factor A is trying to get to its site, has a lot of trouble. At factor B, so the idea is that when factor A tries to get in here, it's pretty bad, but it can get in at some very low frequency, and when it gets in there, maybe it recruits the histone modifying enzymes and puts a little bit of accessibility here, a little bit of nucleosome remodeling, a little bit of acetylation, and that lets factor B have a slightly easier time than it would have if it was just on its own. And then, as you sort of imagine multiple cycles of this, you're essentially cooperatively, that is factors A and factors B, are cooperatively recruiting the nucleosome remodelers and the histone acetylases, and that results in ultimately open chromatin where the two factors are there. And this can be inherited, and so the chromatin facilitates the recruitment of the modifying activity. So the idea is you have two proteins, they cooperatively bind because they cooperatively recruit remodelers that open up the chromatin. And this state can be epigenetically inherited. You can have open chromatin for many generations, but you always need the transcription factors. If you take away the transcription factors, it doesn't work anymore. Okay, and this turns out to be extremely important, because one of the things about human cells is that, and Ellen sort of referred to this, is there's zillions of patterns of gene regulation, and you really can't have the coli mechanism where you have your favorite protein regulating your favorite gene. It's just not possible. And it turns out that this cooperativity is what generates, it's a basic mechanism generating diverse patterns, because it turns out that if you have a single binding site for your favorite factor, A, it won't bind, and B, it won't C, it won't activate. So you need multiple guys to make anything work. And that means that activation is inherently combinatorial. And typically you need at least 3, if not more, of these guys together. And that means that you really now have, a couple thousand transcription factors, you now have, at least in principle, 1 billion possible combinations, and there are other diversity mechanisms. And so then, this is again a very fundamental principle of mammalian transcription for which there is no coli counterpart. So this is what we think is that there is a fundamentally distinct logic of bacterial gene regulation and eukaryotic regulation. And the best way to think of this is the ground state. So what we think of as the ground state is what happens if you have the DNA sequence you need and you have the factors in the cell. What will happen? Well, if you're in E. coli, it's non-restrictive. You have a sequence, you get transcription, okay? So that means that's going to happen all by itself. So that means you don't actually need an activator protein because the polymerase can do it on its own. If you want to regulate things, you can regulate it by having a repressor block it, and you can have an activator help you, but an important concept of the activator is that the activator will only do anything if the promoter is inherently crummy, because if the promoter is really good, you've maxed out and the activator can't possibly help you. So the activator only helps you if the promoter is kind of lousy. Now in mammalian cells, or yeast cells for that matter, because of chromatin, the ground state is restrictive. So you can have a beautiful promoter, works great in vitro with all the basic components. In vivo doesn't work almost at all. And that means you must have activators, okay? Just the basic thing won't work. You need to have activators. And the activators come in two ways. They help recruit chromatin modifying activities, and on its own, that leads to a poised state. Again, a concept that does not exist in bacteria. And that poised state is able to get transcription. Now in the active state, of course, you get the rest of the machinery and you get active transcription. Now you also can have silencing or repression, and this again must mean something beyond the restrictive state. And so you have the recruitment of all these fancy other things that I was telling you about. So the last little bit has to do with epigenetic inheritance. So this is basically the essence of developmental biology, which is why is a muscle cell? And the real question is all the genes in your cells are the same, but your cells are doing different things. And the important point is not only they're doing different things, but they are inherited. So your muscle cells don't become brain cells, they do muscle cells. And it turns out, despite what you might read in various places like trendy journals, that epigenetic inheritance is really mediated primarily by transcriptional regulatory proteins, not chromatin. And there are a couple of examples of that. So one of the early examples is MyoD, which can convert various cell types into muscle cells that are stable. The ones that are much trendier now are the iPS cells, the Yamanaka experiment, where you throw in the right factors and you can pretty much take almost any cell and turn it into an embryonic stem cell. But for those of us old enough, there is actually a precursor to this, which is the work of Mark Potashny, which I think is quite underappreciated these days, basically showing that the inheritance of developmental states in bacteriophage lambda, which are epigenetically inherited, don't require chromatin because we're E. coli and there is no chromatin. Now I blame Mark for this because Mark wrote this great book called The Genetic Switch. And I've hassled him to death because it's a misnomer. It's an epigenetic switch. It involves genes, but the switch is fundamentally epigenetic. It's the same exact lambda, either active or inactive. Okay. So the last little point I'll make is here's a non-bacterial mechanism. This is called position effect variegation. And basically the idea is if a gene is sort of near heterochromatin, depending on what cell you are, it's either off or it's on, okay? And the important point is not only is some cells off and some cells on, but the off cells keep giving you off cells and the on cells keep giving you on cells, but you have exactly the same genes. And they're genetically identical cells, yet they're inheriting completely different states. Cutting a long story short, we basically found that there are reciprocal positive feedback loops that involve the silencing protein, the SERV protein, on histone modifications. And the basic idea is in the off state, the SERV proteins will of course bind and repress, and the SERV proteins, and Lenny demonstrated this, the SERV proteins is a histone deacetylase, and so when it's there, it's deacetylating histones. Well, not only is it deacetylating histones, but the SERV proteins really like to bind to deacetylated histones. So putting the mark in makes it easier to bind, and that makes you have increased SER association, and that inhibits transcription, which means you have less of the active marks put in by the polymerase. And so this repression is blocking transcription so that you have negative marks and you don't have positive marks, and that's a positive feedback loop. In the active version, it's exactly the opposite. The transcription happens, you put in the active marks, now the SER proteins don't like it anymore. So you're flipping from one to the other, and there are no intermediate states here because you pretty much go from one to the other. And the basic idea here is, and this is true for all the known silencing mechanisms, polycomb, HP1, and SER proteins, is the silencing proteins put in histone modifications. They're either enzymes themselves or they're associated with enzymes that put in very specific marks. And they preferentially bind to chromatin with those marks. Conversely, the binding is inhibited by the marks that are put in by transcription. So you have a situation where you set it up by all the transcription factors, but this helps stabilize it. And so this is an important aspect of modifications. So I think I've now told Boris what we've learned, or at least a lot of what we've learned. And so I can imagine if I tell Boris this, I know what the next question will be. So thank you very much.